This is Emma, and my biology role model is Mary Claire King. So let's start it. So she's an American Cancer Society professor at Department of Medicine and Genome Sciences, and she's currently working at University of Washington Seattle. She had her undergraduate school at Carleton College and her graduate program and PhD at the University of California, Berkeley. She had taught at the University of Chile before. So, how did she involve in science? She is born in a Chicago suburb in 1946. She learned the cruelty of cancer when she was still in high school, and a childhood friend died of the disease. Kim became interested in science in the hope to learn enough to prevent and treat such illness. She attributes her interest in science and particularly her groundbreaking work in cancer research in part to that early loss. The interesting fact that she loves using mathematics and statistics to solving genetic problems. So I think Mary Claire King is my biology role model is because she is not only a distinguished scientist who has applied her talents to the good of humanity and provides a clear ideal of what it means to truly impact society through research, but she also applied her genetic skills to human rights work. She used dental genetics to identify missing persons, ultimately identifying 59 children and helping return them to their biological families. She is also a qualified mother. King is a remarkable woman scientist in all aspects and make me admire. So the field that King's involved is mainly genetics. King has focused years of research on cancer, especially breast cancer. Perhaps her most dramatic contribution to date is demonstration of link between breast cancer and a single gene BRCA1. Another example is collaboration with scientists around the world to identify genetic causes of hearing loss and deafness. In recent years, she has developed a deep interest in studying the genetic factor influencing schizophrenia, has discovered evidence that suggests that schizophrenia may be linked to presence of genetic mutations called copy number variations in pathways involved in neural development. So her most recent author paper is published in April 19, 2023, and it's called a paradoxical genotype-phenotype relationship, low level of GOSR2 translation from a non-AUG start codon in a family with profound hearing loss. The main idea of this paper is variable efficiency of Kozak sequences and their embedded start codon may explain congenital profound recessive non-syndromic hearing loss due to Golgi SNAP receptor complex member 2 in an extended consanguineous Peltinian family. Given that known pathogenic allele of GOSR2 are responsible for a recessive neurological phenotype, but with normal hearing, the paradox may be resolved by the combination of unique features of the first codon mutation in the family and differences in GOSR2 binding partners of soluble NSF attachment protein receptor complex in brain neurons. To test whether lower level of GOSR2 protein were due to decreased translation efficiency, use flow cytometry assay to compare mutant plasmids is the technique of this experiment. So this is a figure from the research. So it's about Western blot of GOSR2 translation in transfected cell use in flow experiments with quantification of GOSR2 band intensities normalized to actin and to reference protein levels. Error bars indicate standard errors. That for all mutants plasmid, translation was significantly lower than wild type and that the GeoHSR2 plasmid was translated at a significantly higher level than any of the other mutants. Results from Western blot experiments revealed the same profile. So what's coming next is this coming year, Dr. King and her team will apply rapidly evolving genomic technology to discover complex mutations in DNA that could not otherwise be detected. They will apply this sequencing approach to evaluate families from NYBCS. Dr. King is also exploring inherited genetics variation that's subtly altered expression of estrogen receptor. These subtle effects are not mutation but normal change in expression level of gene that may influence breast cancer risk. So these are the references and the references for images. Thank you.